Hello, can you hear me?
Yeah.
Okay, I would like to thank Doreen and Marcella for inviting me to talk to you today. As Josh has said, at EBI we've been collaborating with Doreen's group at Cold Spring Harbor for a number of years now. Our resource Ensembl plants and Gramene share a common set of data. We work together in preparing new data resources and release in sync. And I think it's a really good way that we have European and American efforts collaborating to produce a consistent global resource of data. So what I'm going to talk today is about getting access to Ensembl genomes by programmatic means. I think some, but not all of these routes are also available through the Gramene site. In either case, it's the same data that's available regardless of whether or not you come to us or go to Gramene. So yeah, here's our homepage, where if you want to look at plant data, here's the Gramene homepage as it's just been presented. One thing about what we do at EBI is we're not only interested in plants, so we also have other sites available offering access to genomes from other parts of the taxonomy. And that means that all the methods for data access I'm going to talk about today can be used to access data from humans, from agricultural animals, from pathogens, from pests, from pollinators, from symbionts, as well as from plants themselves. So the Ensembl web browser is the face of our system, and it's also an important component of the Gramene website, but it's not really suitable for exploring data in very large quantities. If you want to load up a whole chromosome and look at a very large number of genes, you'll find it's relatively slow. So we need to have methods that allow people to analyze data on a large scale, and we have a number of ways in which users can access data programmatically. And most of these utilize standard abstractions for data representation, and this allows us to take advantage of standard tools designed around these abstractions in order to give people access. And so I'm not going to perhaps talk specifically just about programmatic access, but really about various ways of getting data except through the browser, and that will also extend to one or two things which are perhaps not programmatic access in the narrowest of senses. And this is just a slide I normally show near the start of any talk, and I guess the reason I put it in here is you have a summary of the different ways of accessing our data at the bottom of it, which I'm going to go through in more detail now. So if you want to analyze our data in big detail, the most low-tech but most quickest way of getting large amounts of data from our systems onto yours is straightforward by FTP download. All the data that powers our website is available for people to access, and it's available for people to access in a number of different pre-formatted ways. So you can download fast IA files of genomic or protein sequence. You can download annotated files in the format you would find submissions to Ensembl or GenBank in. You can get gene set files. You can get data from our comparative alignment data. You can get data for alignment data. And you can also download the entire database that we run our site off. So if you want to do a really large-scale analysis, the most effective thing is not, in fact, to use any of the methods of programmatic access, nor to use the website, but actually to take the whole data set in a complete form and use it locally. However, we also have ways in which you can program against the data, and one way is to write queries in the database query language SQL. Then our whole Ensembl and Gramene infrastructure runs off infrastructure known as mySQL. It's an open source relational database management system. And so this is a system which gives you a certain standard way of defining a data structure as a specific instance of a general model. And the advantage of doing that is that you can then use a tool like mySQL, which gives you support for defining your data model, for the imposition of integrity constraints and performance optimizations on the model, and gives you a standard grammar structured query language for actually accessing the data, which I guess is what the normal user case for Ensembl and Gramene would be in mySQL, although obviously the integrity constraints are very important to ourselves when we're actually building our resource. So we actually use MySQL as the backend to support most of our pipelines and applications, so it's what we use internally in our own production. But if you want to run your own instance of the databases locally, you can do that yourself. You need to get the database engine from the MySQL site. You then need to come to our FTP sites and download the data and the schema definitions, which we make available. And then it's really very easy. You just have to type a few command line entries in order to establish the database using the data files. And the way our schema is structured is that for each release, for each species, and for each data type, we have a separate schema. So we have a series of schemas that collectively support each species in each release based on the range of data types that we have for that species. And while you can, if you want, and you're going to do a lot of data analysis, do exactly what I've said, install MySQL on your own system, download our data definition files, download our data, and start up our own instance. If you just want to explore the data in a less intensive way, we actually run a public-facing MySQL server outside our own firewall, so you can just connect to our server directly and compose your own queries against it. And so we have a server we use for the non-vertebrate species, but there are similar servers that would allow you to directly connect to our vertebrate databases as well. So what does our schema actually look like? Well, as I said, we tend to actually produce a different schema for each data type we have. So we have separate schemas for our core data, which really means DNA sequence and associated gene models, but also for variation, for comparative genomics, for other features which are alignments or alternative gene models, and for regulatory features. And this statement is in here just to show the naming convention. We have the species, we have the type of schema it is. This is the Ensembl genomes release associated with the database. This is the Ensembl software release, and the last number indicates which version of genome assembly we're actually using. And one way which the whole arrangement works is that there are certain core tables and core identifiers that, as well as being in the core, are also copied to the other schemas to allow queries to be constructed that join together the different data types. Okay, so am I going forwards or backwards? So if you want to know what the schema actually looks like, the schema is quite highly normalized. So normalization is a way you can represent data within a relational database that gives you certain advantages in trying to maintain data integrity. It can come at a performance penalty. So if you look at our core schema, you'll see it's based quite clearly around the central dogma of biology. So in fact, it's not too difficult to find your way around it. We depart from normalization by posting certain keys, like the gene identifier, redundantly across the schema, which allows you to make more efficient queries that involve pulling together data from fewer tables in a way that should improve performance. We have a clear separation of internal and externally facing attributes. So every gene, for example, has a gene identifier, which is the internal identifier within the database that allows different entities relating to that gene to be connected to each other. But there's also a gene stable ID, which is a persistent identifier, which is what you see if you access the gene in the website. Likewise, all our databases, cross-references and methods that we refer to have their own internal name that functions to identify them internally within the database, but there are also display names, which are actually the long form of the names, the correct form of the names, which you will see in the web display, and which may periodically change over time if a database decides to recapitalize its official name or something. There are certain many-to-many linkages within the schema. So, for example, transcripts can be composed of many exons, but an exon can also be found in many transcripts. So there are places where we have linkage tables which link together entities which are found in a many-to-many relationship. Another example of this is how we do cross-referencing. And there's a sort of very generic method for cross-referencing where we link the external database that's being cross-referenced to the XREF, the actual, which is really a join table, then which links in turn to the object xref, which in turn links to the actual Ensembl xref in question. So there's a very standard cross-referencing structure which we can use to cross-reference genes, transcripts, proteins, or any primary object in the database. So you can see that we have one of a number of different tables, and then a standard way in which you can point, say, a uniprot entry. The xref would, so the external DB would define the uniprot database. The xref would then carry the identifier of a particular uniprot entry that was being cross-referenced, and then the object xref table would link that uniprot entry to whichever of the gene, transcript, gene, translation, operon, so on, object types are being cross-referenced. That probably made that sound a little more complicated than it is. If you go into the schema and look at a few actual examples, it's actually, I think, should become reasonably clear. Nonetheless, we still are representing complex data. Here's an illustration of a database schema for Ensembl variation, and it is quite complicated. If you are interested in exploring the schema, I really suggest starting with a core schema, where there's a very clear relationship at the heart of a schema based on the central dogma, and that will be a good way of understanding the general way in which our schema is designed before you go on to look at some of the more complex data types. Here is an example of how you might use our mySQL database. You can log on from any computer to access our public server. You specify the database you want to use. And here is a query which is selecting the public identifier of all genes that actually happen to have a cross-reference which leads to the gene being called waxy. So all our gene names are derived from external databases. So you can find, have display names for the genes through the cross-reference table, and this query would get you all the genes in maize that have been called waxy. I hope that's only one of them, although I don't have the actual answer to this query on this slide to prove it. So what are the pros and cons of using mySQL? Well, it's quick and simple for basic queries. It's very handy for verifying data. If you've done something complex and aren't quite sure if the answer is right, you can go back into the schema and make sure it all makes sense. But as you may have gathered, you need quite a good knowledge of the underlying database schemas. If I'm checking something in Ensembl, this is almost always what I do, but then it's quite natural that I know what the schemas are. The rest of you may have been a little bit intimidated by my previous slides. In addition, the schemas can change between releases as we have to accommodate new data types or we find better ways of doing things. In fact, the core schema is now very, very stable, but some of the other schemas are still more prone to variation. And if you want to do a really advanced query, it can become very complex. Particularly if you're trying to get sequences out of a database, there's some very clever logic you need to do to make sure you actually, say, pull out all the sequences associated with a gene which has been annotated across different contigs in an assembly, and we really don't recommend using it for doing that. So what do we recommend? Well, we recommend using an API. So an API is an applications programming interface. It's a interface for accessing the database which abstracts away the underlying fundamentals. So if you use one of our APIs to access a database, behind the query that you construct, the API is still going to be querying the SQL databases I've just described. But when the schema changes, we change the code between the API and the schema, so the consistent and hopefully simple interface keeps working, and you don't have to worry about the complexity of the underlying plumbing. So the most established Ensembl API is written in the Perl programming language. This was perhaps the most exciting and trendy programming language in the web and in bioinformatics circa year 2000, which was when the Ensembl project started. Now it's perhaps a less fashionable language, but it's still quite a good language for using for data management. We still use it quite heavily internally. And we have a rich and fully featured Perl API, meaning that if you're writing a Perl script, it's easy to just extend your script to make quite advanced queries of an Ensembl database. And it was said of Perl that its strength was that it was designed to make easy things easy and hard things possible. I still really like that mantra as a good theory, I think, that all programming language should aspire to. So our Perl API provides access to genomic sequence, to genome features like genes and translation, to cross-references. It's very fully documented on the internet, so there's an extensive description of all our classes and data models. There's a hierarchical object model within the API, so you can use Perl to write some quite horrible code, but our Perl API works with a nice object model. And the basic principle is that you retrieve first perhaps a slice of a chromosome, then a gene on a slice, and then a feature on the gene. So it's a sort of drill-down model for getting towards particular data you might like. So here's an example. You want to get the gene ID of a maize waxy gene. So it's the same example we saw before. So first of all, you would use this thing called a gene adapter, and you'd speak to the registry, which is a sort of all-powerful module that coordinates your access to the rest of the API. You'd tell it to get an adapter object that will get it genes from the maize core database. You would then try to get the adapter to give you a gene you want. So you'd tell the gene adapter to get you a gene whose display label is waxy. And then finally, you can call the stable ID method on the gene to get that identifier back. So there's some color highlighting in this slide. So these are the objects in the Perl model. These are the methods you can call on the objects in blue, and these are our arguments we're using so that the method actually returns what you want. So we use the API very heavily internally. We use it via analysis and annotation pipelines. The web code also uses it. It's robust, it's reliable, it's well supported. If the API breaks, we can't get a new release out. So almost the fact that we've produced the release is proof that the API is consistent with the database modifications. And it's very, very rare for anybody to report the API isn't working. So what you need to use it, you need to have interpreter of the Perl language installed on your system. You need to have some modules in which, including the API modules itself. We also use some code from an older version of the BioPerl project, which the Ensembl API modules rely on. Obviously, it's a Perl API. That means it's a great way for putting access to Ensembl into your Perl program. So you need to have the ability to code in Perl and to use other APIs in Perl to be able to take advantage of it. Now, for Ensembl genomes, we have a specific extension to the standard Ensembl API, and this includes some additional methods. This is particularly of relevance to our Ensembl bacteria site, where we have, in the latest release, over 20,000 genomes, and we have additional methods for automatically looking up and finding the genomes you might be interested in. For the moment, this isn't yet very interesting for plants, but as the number of different species we represent increases, I think these sort of lookup services will also become valuable. And this is simply an extra module you can download and add to your path if you want to use it. So the Perl API pros and cons. It's powerful, you can retrieve all data, it's stable, it will produce things like translation sequence which aren't directly in the database and which are calculated on the fly. You need to be able to and want to code in Perl to be able to use it though, which in the modern world a decreasing number of people want to do. So we've addressed that by a couple of years ago introducing a new API, and this is the RESTful API. So REST stands for Representational State Transfer. A RESTful API is a particular way of constructing an API. It's constructing an API purely as a URL. So when you go into the internet, you go to a site, you type in a URL, you get to see a page. With a RESTful API, you use a URL and you get back a data object. And it's not just an API via URL, but there are also certain conventions about how that URL is constructed. I think these conventions have emerged out of what the first 10 to 15 years of using the internet established, the World Wide Web established were good practice for the framing of URLs. So good practice in terms of having very clear URLs, very easy to construct, very obvious what data object the URL represents. And because it's all done using the URL paradigm, then it's not tied to any particular programming language that you have to use. So users can access the data simply by invoking the URL. The data is returned in a simple grammar, so using a data representation language like JSON, and then it's relatively easy, whatever language you're coding in, to take the data structure which is returned and convert that into a functional object in your own language. And so one of the nice things about the RESTful URL is you can use it from Perl, but you can use it from Java, you can use it from Python, you can use it probably from Erlang, I guess. So it immediately gives a programmatic access to any programming language that the user is using, providing the user has a method for invoking a URL from that language. So what's needed for it? Nothing. So that's the big advantage over all of the other data access methods I've presented hitherto. I said that in general, you might want to return the object in this simple descriptive scripting language JSON and use your JSON objects to convert them back into the language you're actually programming in. But if you prefer, you can also get your data back formatted as XML. You can also use it to directly download common bioinformatics file formats like FASTA and GFF3. Again, we have it fully documented and examples of actually using it in different languages. So here's an example. You want to get the gene ID of the maize waxy gene. Here you can see the URL. This slide is a couple of years old, so it's when we still had our beta service. Now you no longer need to preface the URL with the word beta. But you can see it just looks like a URL like you might use to access a web page, but it will give you back your data object, and you can then use that in any language you would like. Or if you're working from a Unix command line, well, you can just use any of the standard tools that exist in Unix for retrieving URLs. So the pros and cons: there's no specific knowledge necessary. All languages can send RESTful requests. There's a clean, intuitive API design. The service is still in its early stages. We're consistently increasing the number of endpoints. You can do quite a lot with our RESTful endpoints. If any of you have heard the talks about the variant effect predictor, which have been given elsewhere in this conference, you can use the endpoint to give an identifier of a SNP to Ensembl and ask it what the consequences of that SNP on the gene that encodes it. So you can do quite complex things in the API, but it's still not quite as rich as the Perl API. There are some access limitations. Very heavy data requests will be bounced. If you want to do a massive computational task, the fact is you probably best go of taking FTP and installing the data locally. But for getting access to particular objects that meet particular criteria, the RESTful API is now the easiest and most natural way of doing it. And it's been very popular since it's been launched, and we definitely have a commitment to this going forward. So, access method 5 I don't really need to cover because Josh has already covered it. This is Biomart. So, as Josh said, Biomart is a generic tool to facilitate the design and query of data warehouses. And in this tool, you use a less normalized schema, but it's optimized around certain use cases, and our use cases are optimized around genes and by SNPs. Biomart is still using mySQL as your underlying database engine, but you're building your schema in a different way to give you better performance on certain types of queries. Now, Biomart comes with its own web interface, which has already been presented, but you can also use Biomart to get programmatic access to the data. So I don't need to go over the Biomart web UI again. But at the touch of a button when you do a Biomart query, Biomart results page can be used to generate a RESTful or SOAPy web service, and it will basically auto-generate the code you need to then redo that query on demand from your program. So that's quite nice. You can use the web interface, you can explore the data, you can work out the query you really want to do, and then you can take a chunk of code that you can embed in your program to do that same query again automatically. There is some associated tools associated with Biomart which allow a different sort of programmatic interface. There's a tool called MartShell, for example, which allows users to query the database directly using a command line language called Mart Query Language. I sort of mentioned this on here for the purpose of completeness. In all honesty, I'm not aware of many people using that as a route of data access. Newer versions of Mart also provide a Sparkle endpoint, so would allow you to incorporate your Mart within a sort of semantic web approach. However, we don't yet deploy that version of Mart from Ensembl plants, so we don't offer that facility yet. So, access method 6 is virtual machines. So, I've talked about how if you're a really high volume user, you might want to take the data home and install it on your own computer to avoid taking our servers down or hammering our network. But you might still feel it's rather inconvenient if you have to download our data and establish your own mySQL instance. And if you want a web server running off your own local install, you'd have to establish that as well. But for the past year or so, in fact, it's been very easy to get your own completely local installation. So what we now do is we now release in a virtual environment that includes the databases and the web code and the programmatic APIs. So as long as you have a computer which can support the virtualization environment, it really is very easy to get the whole of an Ensembl system working locally on your own machine. When I say easy, I literally mean minutes. So increasingly, virtual machines are becoming a model of choice in a lot of bioinformatics, in part because you can deploy them on cloud-based platforms, and because the virtual machine brings with it the complete environment needed to run the software, you're insulated from dependencies on libraries or hardware specific to the machine. The one we currently use is a system called OpenBox, which is an open source virtualization platform, and I really think that virtual machines are going to be the way forward for a lot of big computational analysis. So if you're at all interested, check out the web link and get in touch if you have any questions or points of interest. Okay, so that's all I had to say. We're funded by a number of grants, including the NSF, who support us collaborating with Doreen. That's a very positive and constructive collaboration for us. And yeah, I hope we'll continue to work together, and I'll continue to be presenting at these meetings for years to come. And yeah, these are my team. The main people working on plants are Dan Bolster, Arnaud Kervenu, Dan Staines, and Brandon Waltz.
Thank you.